Hello, everyone. My name is Ying. I'm a PhD student at Dr. Stephen Lockey's lab at Queen's University Biology Department. My PhD project uses genomic tools to understand evolutionary history and habitat usage in trilling chorus frogs to infer conservation management. Trilling chorus frogs are species of concern, and some populations are diminishing rapidly in Ontario and Quebec. The main threat is habitat loss and fragmentation. Amphibians are typically vulnerable to that, as they use wetlands for breeding, and wetlands are usually patchy across the landscape. In years of low precipitation or extreme weathers, wetlands can get dried, and populations can go extinct. However, on the landscape with sufficient habitats and good connections among habitats, amphibians might be able to disperse to the adjacent available habitats for survival and breeding, and so the whole populations can persist on the landscape for long term. Therefore, it is critical to understand such metapopulation structure and their habitat usage across the landscape to infer habitat protection and rehabilitation for conservation. Our first question is, to what geographic scale do we find evidence of genetic structure in relation to metapopulation dynamics in trilling chorus frogs? Early this year, I collected over 30 individuals from 9 locations from the land at Queen's University Biological Station, as well as land near Kingston area. I extracted DNA from those samples and used double digest restriction site associated DNA sequencing to generate genome-wide SNP markers. I used both IPARAD and STAX pipelines to call SNPs and use VCF tools to do SNP filtering. I ran analysis including isolation by distance and estimated effective migration surface. What we found is that the effective migration is not uniform across the landscape. For example, in this location, the migration rate is low. This is possible because those two locations are separated by the Apinican Road. So the low migration rates we observe might be a result of habitat fragmentation. Similarly, in this region, those two locations are separated by a busy road, Sydenham Road, and we do observe lower migration rates in this region. In this region that is near KNP Trail, near Kingston, we do see high migration rates, suggesting some population connection in these regions. In the PCA analysis, we do observe some population differentiation in some populations, suggesting some fine genetic structure in this local region. In the isolation by distance analysis, we found higher genetic distance in the among populations with greater geographic distance. From this plot, we can see that there might be some fine genetic structure in this regional landscape. However, because we only have less than 40 samples, and from a few locations, we only have 1 to 2 individuals, we really don't have the statistical power to draw any definitive conclusions. In future, I'd like to collect more samples from those locations, as well as collect individuals from other locations in this local region. I'd like to test the association of landscape features such as forests, roads, or rivers in relation with presence or absence of chorus frogs. I will also use environmental DNA techniques to detect the wetland occupancy of chorus frogs across the landscape. Thank you for listening.